before I put this talk together, I did not realize how well it fits into the general scheme of the conference. So I'm actually quite happy about that. So I would like to show you today insights we gained from a long-term evolution experiment we have been conducting with HIV. I'm showing you the first 3 years, but we are actually in our 5th year now. So for those of you who are into superlatives, this should immediately impress you. But I hope this talk also has something to offer for those who have who are more sophisticated. So this is collaborative work with Karin Metzner, who is at the University Hospital in Zurich. She's an expert on high-throughput sequencing of HIV and does that on clinical samples to detect rare frequency mutations as early as possible, mostly drug resistance mutations. But here we are actually conducting these experiments without any obvious selection pressure coming from drugs or immune effectors. Karin Lehmann contributed quite a bit to these experiments. And on the theoretical side, I'm going to present work we did together with Frederick Bertels, who was a postdoc in my group and is now in Plön at the Evolutionary Biology Max Planck Institute. And a large part of this talk is going to be the work of Eva Bones, who recently graduated with a PhD. So I thought I first am going to give you a little bit of background about our initial motivation. We did not actually intend to conduct long-term HIV evolution. We were coming from a different angle, and I'm going to introduce that. But basically, we wanted to use genetic sequences obtained from different tissue compartments within SIV-infected monkeys to reconstruct the colonization pathway of these hosts. So this was really aiming to get at the population biological understanding of the within-host dynamics of the infection. And then I'm going to mostly spend time on just telling you what insights we gained from these long-term evolution experiments. So I'm going to split it up into the analysis of the first year, where we focused mostly on parallel evolution, and then an analysis of the first 3 years of these experiments, where we are interested in the determinants of HIV evolution in vitro, in particular, to what extent the cell environment shapes the evolution, and also with regard to historical contingencies. So to what degree random mutations in the past shape the evolutionary dynamics in the future. So for the motivation, this came from a review of ours of phylogeographic methods that many of you will know. So these phylogeographic methods we find quite intriguing, because what people do is they take samples, in this case of avian influenza, from different localities. Here you see the yellow pins. And then you reconstruct phylogenies, not just one, but many. And then from that, you can reconstruct the epidemiological history of this epidemic. So this is from a paper by Philippe Lemay that came out over 10 years ago, where they presented this method together with some applications. And what you see here is that they could pinpoint with these methods that avian influenza arose here in Southeast Asia, then it spread locally, then all over Southeast Asia, and then globally. And what you can infer with this method, in addition to where the epidemic started, is the transmission within localities, here shown by the size of these disks, and also the transmission rate between localities, shown by these lines here. And the thickness of the lines corresponds to the transmission rate between these localities. So as I mentioned, we are mostly interested in the within-host dynamics. So we thought this is great, and we might want to apply that to the within-host situation. And if you look at an infection like HIV infection, or also infection with simian immunodeficiency virus, it usually starts at the epithelium, where the virus crosses over into the mucosa-associated lymphatic tissue. That's basically lymphatic tissue close to the epithelium, where it starts to replicate and differentiate genetically, because the mutation rate of HIV is actually quite high. From there, it then migrates into secondary lymphatic tissue, the main compartment where the virus replicates. And there is actually some migration back and forth at rates that we don't really know. We also have not pinned down the replication and diversification rates in these compartments, because usually what we look at is the blood, and the virus in the blood is a composite of all the different compartments in which the virus replicates in the host. So we also don't know about the sampling rates from these compartments into the blood. So we were thinking about applying phylogeographic methods to the within host situation. But actually before we went ahead with this, we thought with Kari Metzner, we should maybe try to validate these methods, that they really give us the answers that we want. So in order to do that, we conceived experiments to assess these phylodynamic methods. And the idea was to set up two simulated tissue compartments in vitro, just by basically taking cultures of two different cell types. And then after pre-adapting the virus to these cell cultures for a few weeks, we thought we would then passage the virus in these evolutionary lines. So we would have lines that would just diversify, and then we would have lines where we transfer virus from here to there or from there to here at predefined rates. And then at the end, we wanted to do some sequencing, do phylogenetic analysis, and see whether we recover the transfer rates that we impose in these experiments. Now, what happened was actually that this burn-in phase that we thought we should apply, because phylogenetic methods are known to work best when the evolution is fairly neutral. This burn-in phase is still going on, basically. So here is the experiment that we actually conducted, which looks very similar, but doesn't have any cross transfers. So we set up two different cell lines, MT2 and MT4, in two replicates each. And then we seeded them with a standard lab strain. These cell lines, I should mention, are derived from the cells of HIV-infected individuals. So they differ. They are standard T-cell lines that are kept in the lab, but genetically they differ by as much as the cell environment differs from person to person during the HIV epidemic. And then we passaged the virus every 3 to 4 days, and every 10th passage we did high throughput sequencing. And I'm going to show you the data up to the 90th passage, which is almost a year. The high throughput sequencing gave us information on what we call majority mutations. That's the main thing we are exploiting here. So what I show here is basically a toy alignment of genomes. The reads we have are longer. They are about 200 base pairs long, but not too much longer than the 10 nucleotides I show you here. And then we focus on majority mutations that are basically defined as new nucleotides that emerge in this experiment that constitute the majority of the nucleotides at a certain locus. Due to the fact that we do high throughput sequencing here, we do not have complete linkage information across the entire genome. So we haven't exploited that in these studies. So the first surprising thing that we found is that if you look at this 1 year of transfer, majority mutations keep accumulating linearly or even over linearly over this time period. We have more majority mutations accumulating in MT2 than in MT4. And what I show at the bottom here is results from a simulation where we factored in the parameters that we think are at play at in this experiment. And we should actually have much less accumulation of these majority mutations if evolution is neutral. I would like to note here that the mutation rate we used here is the mutation rate that Mansky and Thamen estimated. There is a more recent estimate for the mutation rate of HIV in vivo by the group of Rafael San Juan. That is about 10-fold higher. And if you factor that in, we would actually get something that is certainly consistent with the accumulation that we have here. But another indication that the evolution is not neutral in this experiment comes from looking at what mutations accumulate here, because they could be basically different mutations in each of these lines, or there could be always the same mutations popping up. And if you look at this, we see quite a surprising level of parallel evolution. So what I show here is a Venn diagram of the four evolutionary lines. So MT2, the first replicate, the second replicate, MT4 here. And while we have quite a few mutations that are unique to each of these lines, there's quite a lot of overlap, around 40%. There's one mutation that is even reaching majority status in all four of these lines. But there's quite a lot that are shared across across two or more evolutionary lines. And when I say parallel evolution here, I really mean it in a very strict sense. So down to the nucleotide change at the same locus, as you can see. Now, let me just briefly go back to our initial motivation on using phylogeographic methods to look at the population biology of these viruses within the host. So what we have in these experiments, basically by design, is four independent lines. So this is the genealogy, which looks fairly dull, but this is what we have in these experiments. So there is no cross transfer between these lines. If we take the mutations that reach majority and distribute them randomly across the genomes and do a phylogenetic analysis, we can actually recover this independence of the four evolutionary lines. However, if we use the actual data, the consensus sequences that we have at these different time points in the different lines, then the situation is less clear. We have good resolution between the two replicates of MT2, but MT4 is less clear. The phylogenetic tree, due to the parallel evolution, infers quite a bit of common history that if you went on and did a phylogeographic or phyloanatomic analysis, would be wrongly assigned to a migration between the two lines here. So Frederick also came up with a method to use the data in a way that is more consistent with what we know is happening. And that is based on a metric between these alignments, where he basically looked at the frequency difference between the different nucleotides at different loci O between a sample I and J. This is using not just the majority mutations, but basically all the mutations that we have in these alignments, and is also related, I think, to the dissimilarity that Michael Manhard mentioned in the first talk. So if we use this alternative distance measure, then we get nicely resolved evolutionary histories that show that these lines are independent as they should be. So this concludes what I wanted to say about the first year. Let me now move on to the analysis of the first 3 years. So here we wanted to look at the determinants of evolution. And the first thing we wanted to look at was the role of the cell environment in evolution. And just to set the scene, I want to relate this to the concept of fitness landscape. So what I show here is two environments, A and B, and their associated fitness landscapes here shown. Fitness is shown by shade of gray. So here, this area has high fitness. In environment A, this area has high fitness. And if you now start with an ancestor here, then obviously in environment A, it would pick up this mutation, climb up this hill, while in environment B, it would go up here. So these two mutations here are environment-specific adaptations. We are also interested in historical contingency. So here again, if you start with an ancestor virus here in the middle, and if you have two fitness peaks or multiple fitness peaks around this one, then depending on which mutations picked up first, this one or this one, the virus population will climb up either this peak or that peak. Now, the number of fitness peaks around in a fitness landscape is often called ruggedness and is related to the population genetic concept of epistasis, as many of you will know. But we are actually also trying to look at this. Now, in order to do this, we extended the experiment somewhat. So what you see here is these 4 evolutionary lines that I told you about before. And now we are also branching them off. So we switch from this cell environment MT2 into MT4, and also from MT4 into MT2. And then we also have splits where we don't change the cell type. So this gives us some information on the potential role of the environment. And also all of these lines have this stretch of common evolutionary history, which should inform us about historical contingency. So now, as before, we looked at the accumulation of majority mutations. And this is still surprising to us that after 3 years, 300 passages, we still have accumulation of majority mutations almost at a linear rate. As before, we have more of that in MT2 than in MT4. And now, interestingly, the patterns seem to switch over if you switch cell types. So if you go from MT2 into MT4, it flattens out here some bit. If you go from MT4 to MT2, it steepens. You can also include more mutations into this analysis. So you can include mutations that reach over 5% at some stage during these experiments. And then again, you have a difference in the accumulation of these types of mutations across these 2 lines. But now the pattern is reversed. So we have more of these mutations in MT4 than in MT2. And again, switching over gets you closer to the accumulation pattern of the line that characterizes the new cell type. I also wanted to show you some time courses, and we would love to be able to show you these nice Muller plots that we have seen throughout this conference. But as I emphasized before, we don't have linkage information. So what I'm showing you here is the frequency of the different mutations that we look at across these evolutionary lines that might actually be linked. So many of these will be linked and will sit on the same genomes. So what you see here is that in MT2, we have much more green. So green are mutations that reach almost fixation. So at some stage, they are above 99%. So there's a lot of such mutations in MT2, but the dynamics is also wilder. So you see often things going up and then going down dramatically too. We have much fewer mutations that fix in MT4. But we have many more mutations of this purple color that are mutations that are above 5%. So there seems to be a higher mutational supply here. Overall, the patterns of selection in MT4 seem to be more gradual. Now, as also in the analysis of the first year, we can look at sharing, because that carries an important signal for adaptation. And now, because we have different environments and switches, we can look at sharing across both environments and sharing just between different lines of the same environment, MT2 or MT4. So what we find is that most mutations are actually shared between both environments. So the fitness landscapes are not dramatically different between these 2 subtypes. But then we asked whether the degree of mutations, of shared mutations within this one line is actually higher or lower than what we would expect if there there was basically a random distribution of mutations across these evolutionary lines. And in order to answer this question, which goes to the core of how much the evolution depends on the environment, we designed a randomization test. So in this experiment, basically what we have is a table of majority mutations. And here we have the lines in which they occur. So to derive some random or neutral expectation, we just shuffled the lines in which majority mutations occur, but we need to do that carefully. We did not want to change the number of lines in which a majority mutation occurs. And also we didn't want to overall change the accumulation pattern. So you need to do that carefully. But if you do this, then this is what you get. So what we see is that we would expect significantly more mutations shared across both environments than we have. And in MT2, you actually have significantly fewer shared environments, shared mutations than you would expect. We could even put a number on that. Basically, 25 of the 108 mutations here that we observe that are shared only within an environment are likely to be environment-specific adaptations. But we can't say which of these 108 belong to the 25 adaptive ones due to the statistical nature of this analysis. We can also look at the mutations that appear only in one line, either MT2 or MT4. And what we find is that we have many more unique mutations in MT2 than in MT4, than we would expect. So the last aspect we wanted to look at is the role of this mutational history. And here we are exploiting patterns of co-occurrence. So we wonder, do mutations preferentially co-occur in evolutionary lines, or do we have pairs of mutations that never occur together? So we developed these classes of mutations that we call always combinations or never combinations. So always combinations are majority mutations that occur always together, never combinations occur never together. And again, we designed randomization tests to develop an expectation. And here, what these randomization tests do is they take into account the time at which mutations emerge, because early mutations obviously will be in the background of quite a few mutations in the future. So if we do that, then looking at always combinations, the expected number of always combinations is not significantly different from the observed number of always combinations in MT2, but it is in MT4. And in MT4, we basically have more such mutations than we would expect. And this indicates positive epistasis in MT4. Something you don't see from these pictures is that several mutations have always 4 to 5 mutations in the background, which indicates higher order interactions. And then lastly, never combinations. They occur more frequently than expected by chance in both environments. So the observed number is always larger than the expected number. And this translates to about 20% more than expected never combinations in MT2 and 26 in MT4. So this is indicative of negative epistasis in our mind. And this already brings me to my conclusions. So there's 2 types of conclusions. I would mention the first is empirical about this system about HIV. And the first thing that we looked at and that is still surprising is the continuous accumulation of these mutations over this extended period. There's extensive parallel evolution, which confounds our ability to reconstruct the evolutionary history. And what we see is that the majority of shared mutations appear in both environments. So the first order conclusion is that the fitness landscapes of these different environments do not differ dramatically, but still we have enough resolution to find evidence for environment-specific mutations. Also, when we look at these always or never combinations, the fitness landscape seems to be largely smooth. So there's not much, there's not a dramatic difference between the expectation and the observation in terms of these always and never combinations, but we find some evidence for positive or negative epistatic interactions. So I think this is, even though we did not use any strong selectors, as is typically done for such evolution experiments with HIV or other clinically relevant viruses, like drugs or immune effectors like antibodies. I think this is important, an important puzzle stone in understanding the diversification of HIV from one patient to the other, mostly because these two cell lines are derived from two HIV-infected individuals. So they recapitulate the difference in selection pressures and in fitness landscapes that the virus undergoes during transmission. But then we also have methodological conclusions. So what I hope I could show you here is that even though we have high throughput sequences and not much fitness measurements here, and also lack largely information on linkage, by combining patterns of parallelism and temporal patterns together with randomization tests, we can get some insights into these questions. And I think this is important because while I think that the linkage information will become better and better because the sequencing technologies produce larger and larger reads, I think the fitness measurements are the bottleneck in most of this. So even in this experiment, which is not the most complex one, we have thousands of mutations we would have to check, and not just individually on the genome, but we would need to undergo, perform complex genetic engineering to put them together on genomes in all kinds of combinations. So this becomes very soon not feasible. And I don't see that this is going to change anytime soon. So yes, thanks a lot for your attention, and I'm very happy to take your questions.
Thanks a lot for this captivating talk. It was very super interesting. So there are already questions.